This is lecture 5.1 on DNA structure. In this lecture, you are going to learn about the chemical structure of DNA, differentiate between DNA and RNA, and describe how DNA is replicated or synthesized. DNA and RNA are nucleic acids. They have some similarities and some differences. They're similar because they are made up out of nitrogenous bases. They're different in a number of ways. DNA stands for deoxyribonucleic acid. The backbone is made of sugars and phosphates, and the sugar is a deoxyribose sugar. DNA is commonly found in a double strand where you have two sugar phosphate backbones attached to each other by the bases. The bases that you find in DNA include cytosine, guanine, adenine, and thymine. RNA, on the other hand, is generally found in a single strand formation, and that single strand has a sugar phosphate backbone in which the sugar is an oxyribose sugar. The bases that you find in RNA are cytosine, guanine, adenine, and uracil. This next slide helps to summarize basic features of DNA. A polymer made of nucleotides, in which a nucleotide is made of a phosphate part, a sugar part, and a base. The bases that you find in DNA are adenine, thymine, guanine, and cytosine, and DNA is normally stored in the nucleus. The two backbones are held together by hydrogen bonds between the bases. There are 2 hydrogen bonds between thymine and adenine, and 3 hydrogen bonds between guanine and cytosine. A more detailed picture of the structure of DNA shows that we need to pay attention to the sugar phosphate backbones. Because if you think about the DNA as being a sequence of bases, you can have any sequence of bases, A, T, G, C, G, G, A, for example. But how does the cell's machinery know which direction to read the letters? If you compare English to Arabic or Hebrew, for example, in English we read our letters from left to right, whereas in other languages they read the letters from right to left. So it turns out that there's some orientation to the DNA molecule. In this image here, I'd like you to focus on the sugar phosphate backbone. On the left-hand side, next to the adenine molecule up on the top, you see something that says 5' end or 5'. That is the end of the molecule that has got a phosphate group attached to it. And that's referring to a carbon on the sugar molecule. That's the 5th carbon, and that's why we call it the 5' end. If you count the locations of the carbons on your sugar from the oxygen, we normally start it in a clockwise position, a clockwise way from the upwards position. So 1, 2, 3, 4, 5. So it's on the 5th side. If you follow that backbone down all the way down to the bottom, you'll see it says 3' end. The 3' end is the end next to the 3rd carbon. So that indicates that you're next to a sugar. So that backbone runs from sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, if we go from the 3' to the 5' end. The other strand, if you look at the strand that starts next to the thymine on the top, starts at the 3' end. That's the sugar end, and it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and it ends on a phosphate or a 5' end. So these strands are actually running what we call anti-parallel. So they're facing opposite directions. And this is quite important for the cell's machinery because enzymes that are involved in the making of DNA or the reading of DNA have an orientation to them, and they read in particular directions. The width of your DNA molecule is always going to be 3 rings wide. So if you look at adenine, adenine's made with 2 rings and thymine is made with 1 ring. So in total you are 3 rings wide. There are 2 hydrogen bonds between adenine and thymine, and that makes it a stable bond. Guanine and cytosine have 3 hydrogen bonds between them to make a nice stable bond. And once again, you have a situation where you have 3 rings wide. So the rungs of the ladder of DNA are a stable distance. The number of hydrogens between the bases determines the base matching. So adenine will pair with thymine, guanine pairs with cytosine, and so on. What this means is that if you know the sequence of bases on one strand of DNA, you can generate the other strand using those base pair matching rules. So in this example, I've started out with a few for you. If we start out on the 5' end, the matching end would be a 3' end. The adenine would match with the thymine, the cytosine would match with the guanine, and the cytosine would match with the guanine. So I'll challenge you to go back into the slides and see if you can complete a base pair matching rule with a sequence that you make of your own. You can pick any of those 4 bases in any order to make the first strand, then you can generate the second strand using the base pair matching rules. DNA has to synthesize itself in order for you to have the right number of copies of DNA in each cell. Because DNA is used to make proteins, if you have a cell that does not have all the DNA that it needs, it won't be able to make the proteins and the enzymes for that cell to be alive. DNA synthesis happens during a certain phase in the cell's lifetime, called the S phase or the synthesis phase. DNA is described as having a semi-conservative replication process. By semi-conservative, what we mean is that if you have a parent strand that's blue, both the 2 strands are blue, what happens is those 2 strands get separated from each other and each parent strand becomes a template for the new daughter strand. So that means that the new strands of DNA that are created from the old strand each have 1 old strand and 1 new strand. So it's semi or partially conserving the old strand. DNA synthesis requires a lot of different enzymes. One of them is called helicase, and that breaks the hydrogen bonds between the bases. Another important enzyme is called DNA polymerase, and that actually makes the new DNA. Each strand can act as a template for a new strand, and it results in 2 exact copies. So this picture on the right-hand side here shows a DNA double helix with the hydrogen bonds in a pale gray color. Helicase breaks the hydrogen bonds between the 2 strands, and each new strand acts as a template for the synthesis of a new strand. I want you to notice something about the 3rd picture down. When each new strand is being made, it's made in a certain direction. It's made from the 5' to the 3' direction. And at the very end, you end up with 2 strands of DNA, and each has got an identical structure to the original. That was the simple version. There are some additional issues to replication, and a lot of those have got to do with the orientation of the 3' and the 5' ends. DNA polymerase can only work to add nucleotides on a 3' end of a growing strand. So what that means is in real life, one of the strands is made continuously, and that's called the leading strand, and the other strand is made in little chunks. We call that the lagging strand. The little chunks then have to be glued together and they're glued together with yet another enzyme called DNA ligase. DNA is a very long molecule and it tangles quite easily. To control the tangles and facilitate the movement of new strands of DNA into new daughter cells, DNA gets spooled like yarn into little tight packages called chromosomes. It's spooled around a certain type of protein called histones, and that helps to make a stabilized structure. So when you're looking at chromosomes, when we get into the cell replication lecture, when you look at a chromosome, what you're seeing is a tightly bound ball of DNA. It's almost like looking at a ball of yarn. The cell goes through a life cycle. Each cell starts at the beginning of what's called gap 1. Gap 1 is a growth phase. During this point in time, the cell has to make new organelles. It's got to prepare by making sure it's acquired enough materials to have extra nucleotides and extra cell membrane. It then enters what's called the S phase, the synthesis phase, and DNA replicates itself. So you end up with a double your DNA at the end of the S phase. It goes through G2 or gap 2 phase, and during gap 2, the cell prepares itself for division. It enters the M phase, which in most cells is the mitosis phase, and that's where the cell divides and allocates DNA and organelles into new daughter cells. If we follow what happens to DNA through this process, in gap 1, each piece of DNA, if it's folded into a chromosome, forms a single chromosome. Now, when you look at that purple chromosome, you'll notice it has a little pinched part to it. And that's because it has a structure called a kinetochore in the middle. You can think of it as like a little sticky piece of Velcro, and that's the position in which that chromosome can stick to another chromosome. After the S phase, your cell's chromosomes look a little different. They look like a butterfly that's got its wings pinched together. So where those two kinetochores line up, the chromosomes hold on to each other quite tightly. And you've got two identical copies of DNA next to each other. Each copy is then referred to as a sister chromatid. After cell division, those chromatids have been separated from each other. And the process of cell division is the process involved in separating those two sister chromatids. Once the sister chromatids have been separated, they are referred to as daughter chromatids because biologists are weird and that's how we name things. You start out as a sister, you end up as a daughter. Quite funny stuff. Now, we need to talk about more than just one chromosome. In an organism, the complete set of chromosomes, which have the complete set of DNA in order to make that organism, that's referred to as the genome. So in each one of my cells, I actually have two copies of my genome. I got one copy of my genome from my mother and one copy of my genome from my father. So I would be called diploid. However, if I want to reproduce, I need to modify my cells because with reproduction, we need to have fertilization. An egg cell has to get fertilized by a sperm cell. And so if the egg cell and the sperm cell were diploid, what would happen is that the next generation would end up with 4 copies of DNA. And then the next generation would have 8 copies of DNA. And it would become increasingly difficult for the cell to determine which copy to use. So what happens in many organisms is they go through an alternation of generations where you have diploid part of the life cycle and then a haploid part of the life cycle. So in humans, we create sperm and eggs that are haploid, they only have one copy of the genome. In any cell in your body, you can perform what's called a karyotype. It's an analysis that tells you the number and appearance of all the chromosomes in your cell. And this, in fact, is something that you can do when you first become pregnant. You can do a karyotype on or DNA analysis on your fetus. You can do it as an adult. We can do it to do sex determinations on many organisms, from plants to pet birds. This picture here shows you a human adult karyotype. In this case, I'm going to have an extra look at it. It's a boy, so congratulations. You'll notice that there are 23 pairs. Now, in each of those pairs, one of the chromosomes was inherited from the egg, and one of the chromosomes was inherited from the sperm. And in order to have a healthy individual, you need to inherit one of each. So, for example, you might find that on chromosome number 3, there is a gene for something, and you would have a copy of that gene that you got from your mother and a copy of that gene that you got from your father. Now, which version of that gene your body decides to use depends on many other factors. We'll get into those later. But your genome, each copy of it is broken into 23 fragments, and you get one whole set of copies from your mom and one whole set of copies from your dad. And so in your normal skin cell, you would have, if you counted the number of chromosomes in gap 1, you would have 46 chromosomes. One pair of chromosomes is called sex chromosomes, and those can be distinguished from the rest of them, which are called autosomes. Chromosome pairs 1 through 22 are involved in making all of the different enzymes and proteins in the body, and are the same between males and females. The last set, the sex chromosomes, have got genes that are involved in facilitating the development of male or female characteristics. Specifically in humans, females are what we call the homogametic sex, where you have 2 copies of the X chromosome, and males are the heterogametic sex, in which you've got 1 copy of the X chromosome and 1 copy of the Y chromosome. In humans, it's the presence of a Y chromosome that allows the development of male characteristics. There's a particular gene called the SYR gene, and that is involved in testosterone production and masculinization of the human fetus. We will get into this a little bit more later.